Hi, my name is Jacob Gross, and my project is titled Using Mucine Cerebroconnectivity Data to Remap the Mouse Brain. This project uses publicly available information online. You can find the data at connectivity.brain-map.org. Here, you can see a snapshot of the website. Each of these points represents an experiment. Here's how I got started with this project. Over the summer, I worked in a lab at Yale under the guidance of Dr. Alex Kwan. While there, I learned a lot about neuroscience, studying mice, and I wanted to have a project related to that. Dr. Kwan showed me this data, and we discussed potential uses for it and how it might be turned into a valid project. Although Dr. Kwan gave me the idea for this project, I am undertaking the majority of the work myself. Because the data I'm using is available online, I can work on my project from home. If I'm confused about a topic, I'll email Dr. Kwan, but for the most part, this project is largely independent. My rough timeframe for this project is early November to late January. You might be wondering what exactly my project is and what I'll be doing. Well, I'll start with some background. Researchers at the Allen Brain Institute have spent the last several years compiling brain connectivity data. To find this data, they accumulated results from hundreds of different experiments, in which a type of fluorescent bacteria was injected into a specific point of the mouse brain, a specific region or cortex. The path of the bacteria was then tracked and graphed, allowing easy visualization of the connections between cortices, and these paths are now available on the database online. Here's an example of the pathways that you can see. Each one of these lines shows an individual experiment in which fluorescent bacteria were injected, and you can see their path spread throughout the brain. And these are what I'm looking at for my project. Basically, I want to see if pathways from certain regions have similar paths throughout the brain. If they do, this could allow for a more accurate and reliable map of the mouse brain. I'm doing this using statistical analyses, and these analyses will be formed through the use of MATLAB, which is a program that I will teach myself to use over the course of the project. We'll be starting with the secondary motor cortex. I will select 9 experiments, individual experiments to trace, and seeing if there is a correlation between these that leads to something that I can use to support my hypothesis. This data will be analyzed and categorized in different ways in order to see if it does work out to be usable to form a more accurate map of the mouse brain. And hopefully this will be usable in the future.